Chromosomal mutations. These include major changes in the structure of chromosomes such as inversions, translocations, deletions, and duplications in the number of chromosomes. These gross kinds of rearrangements, losses, and gains are cytologically visible in the chromosomes of higher organisms. It results in the changes in chromosome number and in the changes in gross structure of eukaryotic chromosomes. For example, translocation causes genetic changes like Down syndrome. And the Down syndrome is a genetic disorder caused when abnormal cell division results in extra genetic material from chromosome 21. Gene mutation. It is also known as single point mutation. It involves a single nucleotide pair or occasionally a few adjacent nucleotide pairs within the gene or control sequence. They are below the limits of cytological detection as they are causing changes primarily within the genes themselves. Next is about the effects of mutation by mutagenic agents on DNA structure. The two types of point mutation which affect gene expression in different ways include base substitution mutations and frame shift mutations. Base substitution mutation. In this base substitution mutation, a particular base pair is replaced by a different base pair. And it is of two types, transition mutation and transversion mutation. In the case of transition mutation, a purine-pyrimidine base pair is replaced by another purine-pyrimidine base pair. That is AT for GC or GC for AT. So that a purine in one chain is replaced by another purine and pyrimidine in the other chain by another pyrimidine. Transitional mutations occur spontaneously but may also be induced by base analogues, in particular 5-bromouracil, the analog of uracil, and 2-aminopurine, the analog of adenine. For example, transitional mutations are also produced by nitrous acid. It deaminates adenine, whose normal partner is thymine, to form hypoxanthine, which can pair only with the cytosine. Next is about transversional mutations. In this type of mutation, a purine-pyrimidine base pair is replaced by a pyrimidine-purine base pair. Transversions are common in spontaneous mutations. For example, in fact, about half of the mutation of the alpha and beta chains of hemoglobin are transversions. And next about the frame shift mutations. This type of point mutations involve insertion of an extra base pair or deletion of one or more bases from DNA. Due to insertion mutation, frame shift mutation occurs because the normal reading frame relationship for readout of nucleotide triplets in mRNA is put out of register by the mutation. Insertions. Insertions are readily induced by treating cells with mutagenic agents like acridine derivatives. Because of the flat aromatic structure, acridine becomes non-covalently inserted or intercalated between two successive bases in DNA, separating them physically. Therefore, it has a structural similarity with adenine and it can get intercalated in between the successive bases in DNA during replication. When the chain is replicated, an extra base is covalently inserted in the complementary chain opposite to the intercalated acridine. The deletion mutation results from a deletion of one or more bases from the DNA. A deletion may result from hydrolytic loss of a purine base because of high pH or temperature, or by action of covalent cross-linking reagents like alkylating or deaminating agents, which cause formation of bases that cannot repair. For example, the compound proflavin induces deletion mutations. This is the representation of point mutation, and you can see the first one, the wild type gene with a specific base pairing AT, TA, TA, CG, GC, AT, CG, TA, GC, TA, AT, CG, GC. And in the case of transition mutation, you can say one base pair AT is replaced by another base pair GC. Therefore, you can see in the position 4. Here, the CG base AT base pair is replaced by